Okay, chapter 12 is kind of split into a few parts. The first part, we're going to talk about cell division, mitosis, and how normal cell division occurs. And then in later parts, we'll talk about what happens when the cell cycle, I guess you'd say, starts to go awry, go wrong, and give rise to cancer and tumors and things like that. Okay, so for chapter 12, for this particular part here, it's about the cell cycle, about mitosis, and the main idea is that division is essential and it needs to be controlled, and that it needs to know when to start, when to stop, and those sorts of things. Okay, so there's a TOK component here, and what I need you to do is write down your answer to a number of scientific discoveries are claimed to be incidental or serendipitous. To what extent might some of these scientific discoveries be the result of intuition rather than luck? So take a second, write that down in your notebook, and then we'll discuss it in class, okay? So as far as mitosis is concerned, that's basically the division of the nucleus and the splitting up of the contents of the cytoplasm, the cell, kind of into, so that the result is two genetically identical clones of one another. So these cells have to be exactly alike. That's the kind of the most fundamentally important portion of mitosis, is that these cells have to be the same. If they're not the same, then there's some sort of a mutation that's occurred, and of course that's going to be bad. So you want two cells that are exactly like that original one. Okay, and during mitosis, what happens is you've got some sister chromatids, and these things are going to separate. The nucleus is going to divide, and then what happens is cytokinesis. That's kind of the division of the cell. That's going to split the cytoplasm in two and result in two new genetically identical cells that both contain copies of that original DNA, and then that cycle is going to continue to repeat. And of course, this doesn't occur in all cells. It's just dividing cells. Like, for instance, muscle cells don't go through mitosis. Nerve cells, mature nerve cells, don't go through mitosis. A number of other types of cells in your body don't either. Okay, but your skin cells, for instance, your hair cells, things like that, these things are constantly undergoing mitosis and dividing, and you want to make sure that you preserve the fidelity of that original cell that you're starting with. Okay, so if we look at mitosis, you can see here you've got two homologous chromosomes. You've got the the blue one. Whoops, I got to get my pen here. You've got this blue one here on the left. Okay, you've got this red one on the right. And when you go through mitosis, what you're going to do is you're going to you're going to duplicate the cell. And before you do that, you've got to duplicate some of the organelles. You've got to duplicate the genetic material and so forth. And then mitosis will separate these things out. So you end up with basically two daughter cells here. You can see these two from this original one that we started with. Okay, and what you have here, when these things are pinched together, they would be pinched together at the centromere, really at the very kind of center here that you really can't see. You'll see in a later picture here in a minute. But these things are referred to when they're joined together at the center. They're referred to as the sister chromatids, as you can see right here. And then once they go through the cell division, then they become these homologous chromosomes again. So these sister chromosomes or chromatids are going to separate from one another and give the chromosomes in the new cells. Okay, so after anaphase, that's kind of where these chromosomes start to separate out from one another, and you'll see here, we'll go through that shortly. After anaphase, these sister chromatids separate, and now they're referred to as these individual chromosomes again here. Again, these chromosomes are just like these original ones here. Okay, so this is one of the daughter cells, and then this is one of the daughter cells, and it has the genetic complement that you started with. All right, so the genome now, what this consists of is all of the DNA within the particular organism. Okay, and that's important because it contains the genes that are going to control all of the aspects of that particular organism. And prokaryotes typically contain, as you look here, a single circular piece of DNA. In other words, it's kind of like a torus or a donut or a circle. And eukaryotic cells contain multiple linear chromosomes, which is what makes these things different. Okay, so the eukaryotic chromosomes are linear. Prokaryotic chromosomes are circular. They're both double helix. Okay, they're both double helices consisting of the two strands and so forth. And then there's some other kind of aspects of this. The DNA in a prokaryotic cell is typically referred to as naked. In other words, it's not associated with any proteins or any histones or anything like that. There's a lot of extranuclear DNA in terms of these little kind of plasmids that we'll talk about, these little circular pieces of DNA, and we discussed that briefly when we were learning about the cell earlier in the year. They usually contain genes that provide some sort of an advantage to the cell. If it encounters kind of like a rarely encountered, heavy metal or antibiotic resistance is often found on these plasmids. The ability to metabolize certain things and form enzymes to break those things that are rarely encountered down. That's what's on the plasmids. And typically with a bacterial genome, there's one chromosome, whereas with the eukaryotic cell, there's typically many different chromosomes. Okay. So the chromosomes themselves are named because they had the ability to kind of take up some dye. So when you're looking at them microscopically, like you see here, you can see all of these different colors. And early on, when people were studying the cell, cytologists and things like that, when they were providing different kinds of stains, they noticed that these structures started to take up different dyes and take them up differently, and therefore you can get these multicolored types of structures based on the chemical makeup of them. And this is what kind of gives them their name chromosome. And the chromosomes themselves, these are the structures that they're very diffuse during the cell cycle, but during cell division they kind of condense down into structures that make this distribution from the parental cell into the two daughter cells much more manageable, because there's about 6 feet of DNA in each one of the cells, and it would just be like a bowl of spaghetti, more or less, if you were trying to separate that out evenly without condensing it down and organizing it somehow. It would be a mess and it would never work out. So what the eukaryotic cells do is they make these this genetic material much more manageable, in that it condenses down the chromosomes, puts them into a form like you see here that can be easily organized, if you were to do this, but easy to organize from a cell standpoint too, and then divide it up so that each of the daughter cells contain the correct component or correct amount of DNA. Okay. So if you look here, first thing you notice is just the colors there, and that's, hence the word chromosome like we talked about earlier. But chromosomes in eukaryotic cells, we said, are made up of chromatin, and this is really a DNA and protein kind of complex. And again, it's a way to kind of organize the massive amount of DNA that you typically find in a lot of eukaryotic types of cells. Okay, the DNA now within the cell typically contains a few hundred to a few thousand different genes on each chromosome. All right, so there's about 20,000-ish genes in the human genome, and we have the 23 pairs of chromosomes, and then on those chromosomes there's a few hundred to a few thousand genes on each one of the chromosomes. Okay, and these associated proteins, what they're going to do, as we said, help to kind of control the activity of the genes, control the separation of the genetic material during cell division, etc. Okay, so mitosis, there's two main types of cells within mitosis. You either have dividing cells, okay, those are the ones undergoing mitosis, or you have non-dividing cells. So obviously they're not going through mitosis. And then the non-dividing cells, the chromatin is kind of long, very diffuse, spread out. And in a non-dividing cell, the cell is basically in interphase. It's kind of essentially doing its job. And then during the state of non-division, if it's a dividing cell, in other words, if it's not a neuron or not a muscle cell, if it's a cell like an epithelial cell that's constantly dividing, it may be also preparing for division. So there's some things that can be happening there. But typically when it's not dividing, it's doing its job. And then during interphase now, we can break this up, and we'll show you here in a couple minutes. You can break up interphase into different stages, and during one of the stages, the S phase, you can synthesize or duplicate the DNA, and that's preparing the cell to divide to give each of those new cells the correct genetic complement. Okay, and once the DNA has been kind of duplicated, now what happens is the chromatin starts to condense, this DNA starts to supercoil. So you can see up here, the DNA is very spread out, very diffuse, and then now it's going to start getting wrapped around these different types of proteins. And then once you get down here to the end, this is kind of what you have probably in your mind's eye, a picture of what a chromosome looks like. But now it's in a structure that's very easy to move around the cell, easy to divide and distribute to the new cells. Okay. Now this chromosome, once it gets duplicated, you've got these two sister chromatids like we talked about earlier. They contain the identical DNA complement, and they're attached all along their length by proteins. And that centromere, this is the region where they're pinched together, like we said. This is kind of, within the centromere, there's a kinetochore, and that's where these microtubules that we'll talk about in a minute are going to attach so that we can pull these things apart. So if you're looking right here, here's the centromere. We're pointing right towards the center. And then there's a kinetochore kind of right in the middle of that. And these things all along their length, I can kind of adjust the color here, all along their length right here, they're joined up by proteins. That's kind of what's holding these things together. And then in that center again, right here, this is where that kinetochore is. And then this whole structure where they're pinched together, that's kind of the centromere, all right? And then what we'll talk about here now is during cell division, what actually happens, okay? So we'll pick up here in part 2. All right, so if you've got any questions, of course, write them down. And if not, we'll talk to you soon.